Okay, so what Antoine asked me to do is to give a little bit of an overview of a project which is just starting now and which is funded by the INR, where we are trying to build partly on the work which Anael has presented to you, but also on the work which was done in Antoine's lab, where we are taking really the adrenal gland as a model system to address sex-specific differences. And there's a lot of focus here on the stem cell compartment, but we hope that we can then take our findings, which we hopefully find in the adrenal gland, and transfer that to other organ systems. Because what we have seen today is that there's a lot of differences in different organ systems, sex-specific differences in different organ systems. And obviously, it is likely that we will find differences between these organ systems, but also similarities. Okay, so I called that a roadmap to sex-specific adrenal disorders, just because it sounds good. And the people involved in that is, on the one hand, Marie-Christine Chaboissier, Antoine Martinez, myself, and also Serge Neff, who is associated with that. But I'll come to that at the end of the presentation. So Anael has already talked to you quite a bit about this. This is just a different way of representing the sexual dimorphisms which are known to exist in the adrenal gland. If we are taking the mouse, I don't know whether you can see the pointer here, we know that there is a morphological differences, so in adrenal weight, but also in the number of cells which are found in the zona fasciculata. And what Anael has shown you is that there is also a dramatic difference in the stem cell renewal. This is not only found, differences are not only found in the mouse, but also in human. Here are some examples for specific tumors. Unilateral adenoma seem to be very, very frequent in females and only very rare in males. So we have these differences. On the other hand, we heard that this morning as well, when we are looking at how people are doing science, it's actually quite shocking. Especially when you are looking at endocrinology, here the number of papers which are using only males is dramatically, well, it's almost 75%, only maybe 10 or 15% are using females, and in many cases it's not even known. Why is that? Well, it's quite understandable because in the past people took the easy option. Endocrinology, obviously you don't want to be influenced by the cycling in females, and therefore you're using males. But as we have seen today, this has to change, and this is one of the things I think we as a community have to work on. So this slide you have seen as well before. Again, disease prevalence is much more common in females for Cushing syndrome, but adrenal cortical cancer, but also Addison disease. And we have seen the differences in terms of the exon regression in the zona fasciculata thickness, that's in mice. And then there is more and more studies which show that there's a difference in terms of the phenotype. So all of this is obviously done in mice. There's one nice study from Adrien Levassier who showed that when you are doing a YAPTAZ knockout, there is a dramatic phenotype in males, but not so much in females. And this actually is, we see as well with R-spondin 3. So this is a signaling molecule which is important for activating beta-catenin signaling. If that is knocked out in males, and I'll show you an image later on, you see a dramatic reduction in the size very, very quickly in adults. But in females, it is much less severe. Then, on the other hand, when we are looking at constitutive activation of beta-catenin, that's actually work from Antoine's lab again, there seemed to be a predominance in females, and we have another model where we are overactivating our spondin signaling. So this is again acting on the beta-catenin pathway, where we are seeing a similar effect, meaning a very dramatic difference between say, male and female. And then there's also a phenotype in pKa signaling, and I'll just will briefly talk about these two examples. Okay, so this you have seen before. just as a reminder, we have two stem cell compartments. On the one hand, we have a proliferation here within the zona glomerulosa, which is the predominant proliferation or almost exclusive proliferation in males, and that gives rise to these cells which are migrating in, undergo a conversion towards the zona fasciculata, and then they die here, undergo apoptosis. On the other hand, we have the second stem cell compartment, which is sitting in the capsule of the adrenal, and these cells can also be activated, so they are active during development. They give rise actually to the adult adrenal cortex, and then in the adult they are shut down in males, whereas in the females they are actually further contributing to the renewal of the adrenal cortex. So I told you already that R-spondin 3 is very important in that. So R-spondin 3 is expressed in these cells. It signals to the capsular cells out here. R-spondin is out in the capsular cells. It signals here, and then you have a sonic hedgehog Cli1 double paracrine signaling pathway which recruits these cells. So if you knock out that gene, you will see that very quickly these cells, the adrenal cortex is thinning. So this is after only a few weeks. You see this dramatic thinning of the adrenal cortex, whereas the medulla is not affected. And this is actually due to proliferation and not apoptosis, because when you are looking at the apoptosis in a wild type, Anna has already mentioned to you, you get apoptosis here at the junction of the juxtamedullary region here, so between the cortex and the medulla, and this is not changed in the mutant. It's quite striking that you have such a separation between the proliferation and the apoptosis. Okay, so we see that R-spondin signaling is very important for proliferation, and we ask the question, what is happening now when we are ectopically expressing R-spondin throughout the adrenal gland? So, as I mentioned before, R-spondin 3, this is an in situ hybridization, is exclusively expressed within the capsule. Now we use a line which is actually activating R-spondin throughout the adrenal cortex, and we asked what is happening then. And what you see is actually a phenotype which is sex-specific. So firstly, you see an overgrowth. So this is after, I think, 6 weeks or so. Here's a normal female adrenal. This is a female adrenal with this overexpressor. So it's dramatically increased in size. In males, we have also an increase, but it seems to be already a bit smaller. Now, when we are looking over time, we can see that there is quite a dramatic difference. So here is after 3 weeks, after 6 weeks and 6 months. You can see that in females, we have a dramatic increase, whereas in males, the adrenal size is bigger than in the control, but by far not as big as the females. So here again, we have a sex-specific phenotype, which you can also find on the histological level. I here only took as an example the control female. We see this hyperproliferation, this very dramatic increase in the size of the R-spondin 1 overexpressors at 6 weeks of time. Whereas in males, the adrenal is smaller, and you see histologically that there's also a different phenotype. So it's a degeneration of the adrenal cortex. We get this lipid accumulation in there. So this is one example where overexpression can cause a sexual different phenotype. We can also now take a second example where we look at a mutation within the regulatory subunit of PKA signaling. So mutations in that in human patients in the R1 alpha gene will lead to a Cushing phenotype. Whereas when we are doing the same thing, and this is actually Antoine's work, here using an adrenal-specific CRE line, which is causing then a mutation of R1 alpha. So this is an overactivation of PKA signaling. You get a similar phenotype. So what Antoine's lab has shown is that in this particular mouse line, you get also a sex-specific phenotype. This is shown here on this slide, where you can see in females this very dramatic increase in size and in adrenal weight, whereas in males, this is not really visible, at least not an early time point. And when you look at the glucocorticoid production in these mice, you can see that in females you have really dramatic increase in glucocorticoids. In males, it is not obvious, at least not very early on. Then when you are doing gonadectomy, you suddenly see that increase. So it seems to be under androgens. And you can suppress that by giving, after gonadectomy, dihydrotestosterone. OK, so here we have two model systems which show this sex-specific phenotype in the adrenals. So what can be the cause of this sexual dimorphism? On the one hand, and this is now actually more abstract, not specifically on the adrenal, but for all of these sex-specific differences, we have talked a little bit about genetics. Obviously, there could be the sex chromosomes involved. There has been one paper from Bruno Hudry's lab, which is in Drosophila, where they have shown that in an intestinal stem cell, there's actually the sex determination pathway, which is activated. Now, we haven't seen anything like that in mammals, but this is a possibility, and for that reason, I think the sex reversal models, which Marie-Christine has generated, will be a very useful tool, not only in the adrenal, but in other systems, to actually analyze the genetic component in that. Then, certainly in the adrenal, the hormonal component is very important, because we have seen that the phenotype, both for the recruitment of the cells and proliferation, but also in the PKA mutations, can be suppressed by or influenced by DHT, by injection of DHTs, of testosterone. We talked a little bit about epigenetics. Now, this is often actually connected, because when you think about hormonal signaling like androgen receptors, estrogen receptors, they can have a direct effect on the epigenetic landscape of the genome. That has been shown before. So we can think about doing epigenetic analysis on different sexes or in different genetic models. And then what we have heard this morning, that there could be also an influence of microbiota. So there's plenty of different possibilities, and I think a proper analysis is very important. So this is why we have put together a proposal where we are using our genetic models, and we ask essentially three different questions from different angles. So the first thing we want to do is to look at these stem cell dynamics, which seem to be so different in males and females. Looking on the one hand at proliferation, so there we have already quite some data on what is influencing that. We would like to actually correlate that with cell death, and I have shown to you before that, and Annael has actually told you before, that proliferation and cell death, so apoptosis, is separated. However, and we have differences in males and females, and the question is how can you actually, or how can the organism ensure that organ size is controlled properly? Because they can't be, it's very difficult to imagine that they have direct links, that you get more proliferation, then you have to have also more apoptosis. So somehow we need to find a way of actually, or the organ needs to find a way, the organism needs to find a way to control these two different pathways. We talked a little bit about physiology. So physiology in the adrenal gland, where we have these very dramatic differences of turnover, is an interesting question. Why do you have that? What is the point of actually producing 3 times more cells in females than in males? We don't know at the moment. One way of addressing that is to do single-cell sequencing analysis, because then you can actually hopefully identify cell types which are slightly different in males and females. And maybe those cells which are recruited from outside actually have a different makeup, transcriptomic makeup, and produce slightly different hormonal components as well. So while we talked about that, so I don't really have to go into that, what is the hormonal control? How does it influence transcription? Does that also actually has an impact on the epigenetic component? And then we would like to understand how the sex, whether it's chromosomal, whether it's hormonal, is actually influencing the phenotype in our model systems. And as a model system, I have presented to you quickly the overactivation of PKA signaling or the overactivation of beta-catenin signaling. So just to come to the end here of this introduction, the introduction of the discussion. So this is essentially what we are trying to do. We are using mouse models, molecular studies, some in vitro analysis, ChIP-seq and next generation sequencing to address that from different angles and hopefully come up with a clear idea of why and how this differential sexual dimorphism is actually brought about. The people involved, well, this is Antoine Martinez, who I don't think I have to introduce him because he is very well known in the field of adrenal biology, physiology, mouse models, and organizing Firendo. Then we have Marie-Christine Chaboissier, who has generated some of the best sex reversal models and has actually shown that the female pathway is equivalent to the male pathway, that this is really a balance between the two, which is deciding whether an individual is becoming male or female. Then we have Serge Neff, who is in Geneva. He's an interest in sexual development, but over the last few years has really become an expert in single-cell sequencing, and he's associated and will help us with doing this analysis. And, well, I am as well. And maybe we can use that now as a basis to discuss, you know, what how this kind of approach can also be useful for other organ systems. What are actually financial ways of supporting that apart from the RNR? And I was thinking, for example, of a European-wide network, which is grouping together sex-specific sexual dimorphism in different organ systems. I think this would be a really nice way of bringing the community together and also actually raising awareness. In brackets, I'll come to that in a moment, raising awareness to the scientific community that this is a very, very important issue. So I have been a couple of weeks ago at the MRC for a single-cell sequencing evaluation. And the surprising thing to me was that there's actually many of the single-cell sequencing approaches which are performed at the moment, which are not taking into account the sex. So this is quite shocking if you think about all the differences we have seen today. And maybe people who are, well, I'm sure that people who are here in the room don't have to be made aware of, but I think we have the responsibility to also make other people aware of it. And on peut parler en français si vous voulez.